Hello everybody, my name is Chantal Julien and welcome to this presentation. I work at Novo Nordisk Pharmatech. I am the product manager for our recombinant insulin for use in cell culture media. During this presentation, I will introduce you to a DoE statistical approach for media optimization, and I will be presenting two case studies as well where insulin was used in media. So I will begin with a short introduction, also a summary of the function of insulin in cell culture. And then I will present a case study on using the DoE approach for CHO cell media. And I will finally present two case studies using CHO cells and HEK-293 cells. So a few words about who we are. At Novo Nordisk Pharmatech, we are located in Denmark, and we are 100% owned by Novo Nordisk, which is the global leading manufacturer of therapeutic insulin. At Novo Nordisk Pharmatech, we are a supplier of ingredients to the pharmaceutical and biopharmaceutical manufacturers worldwide, including recombinant insulin. So what is insulin cellular functionality? In vivo, insulin is a major metabolism regulating hormone. It is secreted by beta cells of the islets of Langerhans in the pancreas. The major function of insulin is to maintain low blood glucose levels. In vitro, insulin functions as a growth factor. Insulin is actually a member of a family of growth factors, including the insulin-like growth factors IGF-1 and IGF-2. Insulin regulates cell growth and differentiation, cell survival, and cellular glucose uptake by modulating transcription, DNA synthesis, replication, and stimulating protein translocation. Different intracellular signaling pathways are initiated by insulin binding to its receptor, such as the activation of PI3 kinase AKT signaling, triggering glucose homeostasis with glucose transporter GLUT4 translocation and fusion with the plasma membrane, and the activation of RAS-ERK pathway with activation of transcription, DNA synthesis, and cell growth. Now, the cell culture media industry has advanced over the years, from serum-based media to animal-derived component-free media. A continuing trend is to shift towards defined systems to support manufacturing quality requirements of cell therapy, gene therapy, tissue processing, vaccines, and recombinant protein. Today, the most extended practice in the culture of mammalian cells is the use of commercially available, chemically defined CD media. These media are optimized to support cell growth and recombinant protein production and are usually very complex. However, their composition is usually proprietary and unknown for the user. When used for a specific cell line, CD media may not support all its specific requirements, and therefore additional supplementation with other compounds at optimal concentrations may provide substantial improvement in cell performance. There are different approaches to media optimization. The case study presented here was conducted in a partnership with Dr. Francesc Godia at the University of Barcelona, and we used a CHO cell line. CHO is still the preferred mammalian cell host for production of recombinant protein, including mabs. Media supplementation is generally addressed by means of a one factor at a time or classical design of experiment approach. But these techniques may not be efficient in initial screening phases, as they are time-consuming and costly. In this study, a DoE statistical approach was used to achieve the best performance in increasing both cell growth and Mab production through supplementation with animal-free recombinant compounds. 8 components, as you can see on the slide, were selected according to their capacities to improve cell culture conditions in a CHO cell culture system. A Plackett-Burman design and a Box-Behnken design were used, and synergies between compounds could be detected with a reduced number of experiments by using this method compared to more conventional design. And this led to a fast and efficient screening and concentration optimization phase. The first part of the work consisted of a folded over PBD, so Plackett-Burman design, and was used to detect possible synergies between different compounds in the screening phase. PBDs are developed to eliminate factors having no or negligible effects over the responses. This means that only main effects are considered without detection of factor interactions leading to potential incorrect decisions. So this would be the traditional PBD, which is shown on graph A. On the other hand, a folded over matrix PBD, here in this example on graph B, we have a 24-run folded over PBD, improves the detection of factor interactions in the screening phase of a full DoE approach. Growth kinetics were followed every 24 hours during 8 days. That allowed to determine the maximum viable cell concentration for each condition tested. The 8 compounds were screened at 2 levels, a low level, meaning no additive, and a high level that was defined based on toxicity experiments. The classical approach showed transferrin was the only factor affecting cell growth. That would be on graph A. While the model containing interactions on graph B showed insulin and 2 significant two-way interactions between transferrin and insulin and transferrin and twin AT. Therefore, recombinant transferrin and insulin and twin AT were chosen for subsequent concentration optimization using the Box-Behnken design to optimize the concentrations. So in this phase, in the Box-Behnken design phase, the range of insulin concentration was reduced since no improvement in cell growth was observed beyond 2 milligrams per liter of insulin. And 3D plots were constructed for visual analysis of the response, illustrating different combinations of insulin, transferrin, and twin AT. The highest level of cell concentration were obtained with a high level of transferrin or with the highest level of insulin and a moderate level of transferrin. That is seen in graphs D to F. And as the transferrin concentration increased, the optimum condition moved from higher to lower insulin concentrations. And that is seen in graphs A to C. The optimum with transferrin was achieved with a very high level at 57 milligrams per liter. And this was considered difficult to be adopted in practical terms due to the high cost of transferrin. And therefore, 21.3 milligram per liter of transferrin, 2 milligram per liter of insulin, and 1.8 times of twin AT was selected as being the optimum condition. And under this condition, the predicted maximum viable cell concentration was 9 plus minus 1 million cells per milliliter. And for the full data and statistic analysis, you can refer to a published paper called A Statistical Approach to Improve Compound Screening in Cell Culture Media that was published in Engineering in Life Science in 2018 by Professor Godia's group. Now, following the DoE approach, optimal conditions were validated against a negative control, CD media without supplementation, and cell growth and cell productivity were measured. The triangle in light blue, the line with the triangle in light blue, shows that cell growth in the CD optimized media versus the dark blue line, which is the negative control. And the light blue circle line shows that mab titers in the CD optimized media was better than the one with the pink triangles, which was the negative control. And under these optimized conditions, the viable cell concentration reached the predicted 9 million cells per milliliter in a Mab titer of 120 milligrams per liter. And to summarize, the sequential use of this modified Plackett-Burman in combination with a Box-Behnken design led to a 1.5-fold increase in cell growth and a 2-fold increase in a Mab titer in suspension batch culture. As a follow-up to this study, the effect of insulin media supplementation was tested in 3 types of commercially available CHO-S CD media that are called here CD1, CD2, and CD3. And that was to compare cell growth and cell productivity in a suspension batch CHO cell culture system. The black diamonds show the negative control CD media without insulin, and the green and light blue lines represent respectively 1 milligram per liter and 5 milligram per liter of insulin supplementation. And already after 48 hours culture, a significant improvement in cell growth was achieved with the addition of insulin. And in all 3 CD media, differences in cell growth peak at around 120 hours were observed before a viable cell density drop. And this is due to the saturated culture conditions. The same CD CHO media were tested for Mab productivity with and without insulin supplementation. And here we achieved up to 66% increase of target protein production with 1 milligram per liter of insulin supplementation. A second case study was generated in collaboration with the National Research Council in Canada, conducted in the lab of Dr. Aziza Mansour. The aim of this project was to test the effect of insulin supplementation in HEK-293 cell growth and on virus production. So an in-house suspension cell line was utilized in the study. The in-house serum-free 3F61 cells have previously successfully been used for the production of influenza virus, virus-like particles, lentiviral vectors, adenovirus, and adeno-associated virus. Here they were tested in a commercially available CD HEK-293 media called CD1 and an in-house CD HEK-293 media that is called CD2 here, with and without insulin supplementation. Insulin additions were performed every 72 hours at 2 different concentrations, 10 and 20 milligram per liter of insulin. The dark blue line here represents the negative control with no insulin. And in CD1, the cell density remains below 1.5 million cells per liter. With addition of insulin, on the other hand, viable cell density reached about 6 million cells per milliliter. And there were no significant difference between the two concentrations of insulin. In the second set of experiment, it was performed on CD2. With this media was developed specifically for this cell line. So with or without insulin, a cell density of nearly 6 million cells per milliliter is achieved. However, this level can be reached about 3 days earlier with the addition of insulin, thereby accelerating the process. The effect of insulin supplementation on CD media was also tested on influenza virus productivity. Infection was performed with a multiplicity of infection, an MOI of 0.01. That means one viral particle for every 100 cells. H1N1 Puerto Rico 834 serotype was selected, and 3 different concentrations of insulin ranging from 5 milligram per liter to 25 milligram per liter were tested. Insulin was added at the time of infection, together with the addition of trypsin. The viral titer was measured by immunoblood assay against HA epitope. Also in this case, insulin supplementation led up to a 1.6-fold increase of viral production compared to the control CD media. Lastly, should you need recombinant human insulin, animal-free insulin for your cell culture media, the recombinant insulin that we offer is manufactured by our mother company, Novo Nordisk, and it is manufactured according to the highest available standards on the market, the GMP standards, and it is analyzed in compliance with the European Pharmacopoeia and the US Pharmacopoeia. And we also offer a complete documentation package. You are welcome to contact us for more information, or of course to visit our homepage at novonordiskpharmatech.com if you want to get more information. Thank you very much for your attention today, and please do not hesitate to write with your questions. We will be happy to respond. Thank you.